Hello everyone and welcome to the continuation of Chapter 2, which is on chemistry. And we just have our section on organic chemistry left, and I'm going to do my best to make this short and sweet and focused on what you need to know for your exam. So we'll be covering our macromolecules here. We have carbohydrates, lipids, and proteins, and we're also going to talk about some nucleic acids toward the end. So this is more of a summary slide. The only thing I want to draw your attention to here is that our carbs and lipids are going to both be composed of carbons, hydrogens, and oxygen. And our proteins have carbon, hydrogen, and oxygen, but we're also going to add in nitrogen and sometimes iodine. And our nucleic acids will also have carbon, hydrogen, and oxygen, and we have added in the nitrogen and phosphorus here. So we'll go through some of the functions as we discuss further details. This chart here is showing you some of the major functional groups that belong to our organic compounds. We'll be discussing some of these other ones in our lectures in the future. The ones right now I want you to focus on is the amino, which we'll get to when we talk about proteins, and also phosphate for when we discuss ATP. So first we'll start off with our carbohydrates, and the simplest sugar that we can discuss here is monosaccharides. So mono meaning one and saccharide referring to sugar. And you'll see that these compounds are either made up of 5 carbon sugars or 6 carbon sugars. And so when we see a ring like this, each point counts as a carbon. So we've got 1 carbon here, 2, 3, 4. So we've got 4 carbons here, and then we have our 5th carbon up here and our 6th carbon over here. So this compound together would be fructose, and I'm not going to have you memorize the formula or the ring structure for each of these, but rather I'd like for you to be familiar with the names. So we've got fructose, glucose, and galactose, and all of these would be single sugars. And you could see based on where we have our carboxyl group is going to change the type of sugar that we have. So where we're going to find more of those 5 carbon sugars are parts of ATP and DNA and RNA. So really the deoxyribose and ribose itself, but we'll look at that at the end of this lecture. So when we combine these simple sugars with another simple sugar, we are going to get something called disaccharides. Di meaning 2, saccharides again referring to the sugars. So we have 2 simple sugars that get combined together. Look at this, we are taking 2 hydrogens and an oxygen, and we are losing that, so this would be a dehydration reaction, right? And that is helping to link the sugars together, and we now have sucrose. Other examples of disaccharides would be lactose and maltose. And then when we have more than 2, we are going to call that polysaccharides, and these are going to be longer chains of sugars added to one another. So the one I really want you to know is going to be glycogen, and you can see there are many chains of glucose linked together, and animals keep this as storage for sugar when they need it. We know that we will get starch and cellulose formed for plants in order to store their sugar that they need. And then this is just a chart that summarizes our role of carbohydrates within our body. I won't go through it again. And that brings us to our lipids. So when we look at our lipids, we have a strip of glycerol, and then we have fatty acid chains that extend on out. When we take water away from this molecule, we have dehydration take place, and enzymes are helping this reaction move over toward the right, and we link it together to get something called triglycerides. And things that our lipids are going to help us do is to protect. So we know our subcutaneous fat helps with protection and cushioning and absorption, and it helps to insulate us, not only our entire body to insulate body heat, but it'll also help to insulate our nerves as well, so that we get nerve impulses moving through faster. And we can also store lipids as a fat for energy source when we need it. So we can break this down into three types of lipids. We have our saturated fats, our unsaturated fats, and trans fats. So what does saturated mean? It means that this entire carbon chain is saturated with hydrogens. I don't have any double bonds in here, so we get a really rigid structure. When you look down here at unsaturated fat, this means that we have at least one or more double bonds within our chain. So we see double bond here, as well as over here. And this is more of a relaxed structure to our molecule, and this is great because it doesn't stick to our blood vessels and cause plaques. And I wanted to show you a picture of what a trans fat looks like. And there are natural trans fats within animal fats. So those aren't so terrible for us, but the artificial ones that we create, like in margarine or vegetable oils, they have a double bond in here, and the trans term tells us that these hydrogens are angled in opposite fashions from one another, whereas an unsaturated fat where we have a double bond here, the cis fashion tells us that the hydrogens are both on the same side. So still not the best always, but the trans fat is definitely worse for us in causing cardiovascular diseases and disorders. So that brings us to our phospholipids, and we are going to find the main place where phospholipids are found are in the plasma membrane of cells. And the way they are set up is that we have this polar or hydrophilic head, which is going to basically really love aqueous solution, so a water type of solution, which is why it's called hydrophilic. So this faces the outside and the inside of the cell. I should have put a picture of what this looks like, but we'll be visiting this in chapter 3 again. And then we have the nonpolar or the hydrophobic tail, which is made up of fatty acid chains. So I like this image over here because it shows you a saturated fatty acid chain, so you can see no double bonds here, and that's why it's nice and straight. Whereas the other chain, it is unsaturated, and so we have a double bond right here, and that causes a little kink to its the rest of the tail. Okay, I decided to get rid of one slide for you all, so don't worry about this slide here. And then I think the last lipid we're going to talk about are steroids here. All of the steroids we'll discuss are derived from cholesterol. And basically we have 4 carbon rings linked together, and by changing some of the other components attached to those rings, or the rings itself, we can change to hormones like estrogen or estradiol, testosterone, and we can even change over to a bile salt. So we know that our hormones are going to help to regulate our body. Bile salts will do that as well, and we'll get it more into detail in our endocrine system and digestive system on those. And here's a summary of all of the functions that lipids do for our body. So I'll let you read that on your own. If you have any questions, please reach out. And we get to our proteins finally. So we cut out little amino acids in class. Those are our building blocks of proteins. So when we were taping one amino acid to another, we were binding them with tape, but what happens in reality is that we are going to take out a water molecule. So once again, we have a dehydration reaction here, and that bond that gets created is going to be a peptide bond. So when we get many amino acids coming together, about 10 or so coming together, we are then going to call that chain a peptide. When we get more than 10, we would call that a polypeptide chain. And then, of course, as we get these polypeptide chains all coming together and folding in different fashions, we will get a protein. So I wanted to give you an idea of what these amino acids look like. Here are 20 of our common amino acids. So you'll see that all of them have a similar structure. We have our main carbon, a hydrogen, our nitrogenous group, or amine group we could call it, and then our carboxyl group. And the portion that's shaded is going to be the R group. And what that means is that this is variable. So it's going to change with every different type of amino acid that can be made. So I won't have you memorize these different amino acids. That's more for a biochemistry course. But know that these amino acids will link to one another with peptide chains. And so here are the roles of proteins in our body. They do a lot for us. We'll be discussing a lot of these functions in chapter 3 as well. So let's review the protein structure and how a protein comes to be. So in class, we added all of those amino acids and created a long string chain of the amino acids. So we're going to call this a polypeptide now. Now the secondary structure is when we start to fold our amino acids into either beta pleats or just pleated sheets or alpha helices. Now when we do that, we are creating some hydrogen bonds. And remember, that is some intermolecular bonding here so that we can keep this conformation of our secondary structure. When those secondary structures come together, we get our tertiary structures. So here we can see an alpha helice. We could see these are representing some beta pleating over here and here. And so when we're folding these, we also have some other interactions take place, like for example, disulfide bonds that holds this tertiary structure folded up the way that it is. And so when we take this subunit, this tertiary subunit, and combine it with another subunit that has gone through the same process, we can create our quaternary structure, and now we should have a functioning protein. And the last topic, well, I should say last topic because we're going to talk about ATP and deoxyribose and ribose. But next we'll talk about enzymes. And we already mentioned enzymes. We said they help to lower our activation energy. And what an enzyme does is it basically has an active site in which our reactants are going to fit into the specific space it's meant to. Usually when that happens, we have some type of conformational change, and that makes it active so that we can cause the reaction to move along faster or more to the right. Also, these enzymes have really specific names. Most of them are going to end with an ACE to let us know that it is an enzyme. So an example would be peptidase or lipase, and the name usually gives us an idea of what that enzyme is going to do. For example, lipase will help to break down lipids. And some will even have cofactors or coenzymes. So when we have an active enzyme, we can combine it with a coenzyme, and that will help to activate the enzyme once it comes into contact with its substrate, so that the enzyme can work correctly. So quickly, let's talk about some nucleic acids. Most of you already know much about DNA and RNA, but this is going to be a quick little review. So here is our deoxyribose, and here is our ribose molecule. And you can see that they are pretty similar to one another. We are going to use deoxyribose, of course, in our DNA, and ribose will be used within our RNA. And the differences that we're going to find between these two molecules is that deoxyribose will have a hydrogen here on its third carbon, and on ribose we will have a hydroxyl group. So very minor difference, but enough to cause a big difference, if that makes sense. And attached to deoxyribose will be a phosphate group and a nitrogen base. And our nitrogen bases are listed here for us. Here are our pyrimidines, which is cytosine, thymine, and uracil. And our purines are guanine and adenine. And so all of them are going to be found in both DNA and RNA, except for thymine will only be found in DNA, and uracil will only be used in RNA. And so what do we use DNA for? Well, it stores our genetic material, which we pass down to our offspring, and we are going to have 2 strands of these nucleotides. So let's take a look at what this looks like on the next slide. Looks quite complicated, but here we have the backbone of our DNA, where we have our deoxyribose molecule and the phosphate. So that connects over to our nitrogen bases. Here's our thymine, and that will always be attracted to adenine. And then our guanine will always be attracted to our cytosine. And notice that we have these double bonds in place here with our T and A, and over here we have triple bonds with our G and C. So again, we are talking about DNA, so there's no uracil found here. Here is RNA, and we only have one single strand that makes RNA up. We said that it's going to have ribose within its strand, and then we have our adenine, guanine, uracil now instead of thymine, and cytosine that will make up the nitrogen bases here. And so I quickly want to walk through the process of protein synthesis. You're going to go into more detail in your microbiology course, which I believe is next quarter. So this is just a little overview, but we take our DNA double strand here, and we are going to replicate it and go through a process called transcription. And transcription will read our sense strand so that we can make a copy of that using the codons. So you'll kind of see that your mRNA strand will be pretty much exactly the DNA strand here, except we change the thymine to uracil. And so after we go through transcription, we are going to go through translation. So our mRNA strand has these tRNAs that will come in with an anticodon. So we are reading the opposite of each of these, and that anticodon will code for specific amino acids. So when you guys cut up your amino acid pieces in our class, you saw that there were a bunch of letters attached to it. That's what those letters represent. They're the anticodons on the tRNA that code for the specific amino acid. And so a ribosome will receive those tRNAs and will basically assist to link up the amino acids. So methionine with glycine here, we get a peptide bound here with leucine, and so forth. Peptide bonds all around until we create our long peptide strand. So the last thing we'll mention is adenosine triphosphate, or ATP. We discussed this in class, that it is made up of a ribose molecule, adenine, and then based on how many phosphate groups we have either adenosine monophosphate, so that would be one group, adenosine diphosphate with 2 phosphate groups, and adenosine triphosphate, which would be the 3 phosphate groups. So every time we break off one phosphate, we get energy that is released. Now keep in mind that not all of that energy can be utilized, and so some of it will dissipate as heat. So that is it. Please let me know if you have any questions at all, and good luck on your upcoming exam.